Scale up of aeration and agitation. As aeration and agitation dominates the study of scale up, as we have seen in the earlier lecture, more concentration has to be focused on the adjustments of the parameters of aeration and agitation. The solution to the scale up problem is in threefold. It can be obtained in three stages. First one is the identification of principal environmental domain, that is the environmental parameter or the environmental area affected by aeration and agitation in the fermentation. For example, oxygen concentration domain, shear condition domain, or bulk mixing domain. So first step is the identification of this domain which is affected by aeration and agitation. Second step, the identification of process variable or the variables which affects this identified environmental domain. And the third step, the calculation of the value of process variable to be used on large scale, which will result in replication of the same environmental conditions on both the scales. That means first step involves the identification of the domain. Second step involves the identification of the process variable which is related to that domain. And third step involves the modification or the changes according to the calculated value so as to obtain the same conditions as that of the earlier scale in order to get the same productivity. The effect of process variables on mass transfer or mixing characteristics. So, what are these process variables and what are the characteristics which are affected are shown briefly in this table. Process variable, if it is power consumption per unit volume, then the characteristic affected is oxygen transfer rate. If process variable is volumetric air flow rate, then it is related to oxygen transfer rate. If process variable is impeller tip speed, then the characteristic which is going to be affected is the shear rate. Shear means the damage to the cells, so on and so forth. Now, for example, if according to these threefold solution, if dissolved oxygen concentration is identified as the most overriding, means most important or critical environmental condition, then that will be the domain. Then power consumption and power consumption per unit volume and volumetric airflow rate per unit volume should be maintained constant on next scale also. How we have come to this statement? Based on this oxygen transfer rate. So, what are the process variables related to oxygen transfer rate? Power consumption per unit volume and volumetric airflow rate. So, here, if dissolved oxygen is that overriding parameter, then power consumption per unit volume and volumetric airflow rate has to be modified accordingly. However, as a result, the parameters will not remain same in large scale, and therefore neither will the environmental factors which they influence. This means that if we keep dissolved oxygen concentration, if we keep on concentrating on dissolved oxygen concentration, and if we keep power consumption per unit volume and volumetric airflow rate conditions constant, then the other variables are going to be changed, or other variables will never behave as normal, and these values will be affected. The parameters may decrease or increase. Let us see an example of that. In this example, old shoes example, where there is 125 fold increase in the scale is represented in this table. You can see the first column is criterion used in scale up is power by volume, volumetric airflow rate, and impeller tip speed. Second column is the effect on the operating conditions on the large scale. These values are large scale values divided by small scale values. So if you concentrate on this second subcolumn, that is P by V. So here the value of P by V in the first row is 1. This means that the power consumption per unit volume, the value are same on small scale as well as on large scale. Therefore, the ratio is 1. In flow per minute per volume, the second row shows value of 1. This means that here the flow rate is constant, means the value of flow rate at small scale and at large scale are same. That is why the ratio is 1. And NDI means it is impeller tip speed. So at the last row it is showing 1. That means here the impeller tip speed at small scale and large scale are same. So here what we are going to understand is, if we keep a particular parameter constant, that means if the ratio is 1, then the other variables are changed. They behave abnormally. Say for example, if power consumption per unit volume, you see the first row here. So if the power consumption per unit volume is constant, that is 1, then you can see that the flow has decreased and impeller tip speed, that is NDI, has increased. In the second row, if the flow characteristics are constant, then there will be tremendous increase in power consumption per unit volume, that is 25, and there will be tremendous increase in the impeller tip speed, that is 5. In the third row, if impeller tip speed is kept constant, then the flow rate will decrease and also it will affect the power consumption per unit volume. This table shows that we cannot keep only one condition constant. So at a time, the modifications has to be done in several other parameters also. The most important environmental domain affected by aeration and agitation in most of the fermentations are oxygen concentration and shear conditions. So therefore, the most widely used scale-up criteria are the maintenance of KLA and constant shear conditions. So KLA is related to oxygen concentration, and another parameter is shear condition. So these two has to be maintained properly in order to get a good progress of fermentation. So constant shear conditions. Constant shear conditions means on small scale as well as on large scale, the conditions should remain same. So constant shear conditions may be achieved by scaling up. Scaling up means it is the study of parameters. So scaling up on the basis of constant impeller tip speed, and constant KLA may be achieved by scaling up on the basis of constant power consumption per unit volume and constant volumetric airflow rate. Now the operating variable that dictates the constant power consumption in geometric similar vessel or in the fermenter design is agitator, the speed of agitator. So speed of agitator, it is related to power consumption. So the agitator speed on large scale is then calculated from the correlations. So all these are the sorts of calculations. We are not going into the too much details, but this leads to the calculation using different equations. So what are these correlations? These are the correlations between KLA and power consumption, and these are the correlations between power consumption and operating variable. So using all these relations and calculations, finally what we calculate is the speed of agitation. That is the final goal. Now exactly what is this KLA? We need to remember this. Definition of KLA is volumetric mass transfer coefficient. It is the measure of aeration capacity of fermenter, and this KLA, very important, it is affected by aeration rate. KLA is affected by agitation speed. KLA is affected by impeller design. KLA is affected by culture broth or the viscosity of the broth, called as broth rheology. So volumetric mass transfer coefficient, it is transfer of what? Mass means everything which is present inside the liquid medium. What is present in liquid medium? It is indicated by volumetric means it is in a given liquid volume. Transfer of mass means transfer of nutrients, transfer of oxygen inside the producer organism's cell. It is called as volumetric mass transfer coefficient. So obviously it is affected by these different parameters which are related to aeration and agitation. Aeration and agitation, it is a combined process, it is a combined function in order to maintain the continuous rate of aeration. It has to be uh, get assisted by agitation. The optimum KLA, means required KLA, is determined in small scale fermenters and it is employed on next scale. So the KLA value is calculated on small scale, that will give you the higher productivity, and we'll try to maintain the same KLA value in next level fermenter by the modifications of different process variables. So same KLA, it can be obtained in different size fermenter. This is very important, by adjusting the operational conditions. So the quantitative relationship between KLA and other operating variables is very important for the calculation of values of the process variables required to obtain the same KLA. That is all about the scale up of aeration and agitation, how it is done and how it is calculated at small scale and how it is applied to large scale. Then second part is scale up of broth rheology. That means flow characteristics of the culture broth has to be studied in detail, because flow characteristics changes from when we increase the scale. When we go from small scale to large scale, the volume increases and the broth characteristics, cultural broth characteristics, the viscosity of the broth, it also changes. Therefore the KLA has to be calculated considering the broth rheology. So rheology means the flow characteristics of culture broth. So fermentation broth, it is a mixture of several things, and it may become viscous, or the flow characteristics of this fermentation broth, it changes, it keeps on changing during the fermentation. Why it changes? It changes, the viscosity changes, the flow characteristics changes due to the composition of original medium. That means if the original medium contains certain jelly substances or certain solid matter, then the viscosity of the broth is increased or it is affected. Say for example, it contains starchy substances, then the viscosity increases. So initial composition of the medium is responsible for rheological properties of fermentation broth. Then once the microorganism starts growing in that, they will degrade. Say for example, starch, and the viscosity of the broth will again decrease. So modification of broth by growing microorganisms. On the other hand, certain microorganisms may produce certain viscous substances. That may be the point to be considered. Then the broth rheology is affected by biomass concentration. So biomass may be of different types, like biomass morphology and biomass concentration. So biomass morphology means whether the organisms are unicellular or mycelial organisms. So depending on that, the broth characteristics will fluctuate. Say for example, in case of unicellular fermentations, the broth may not be that viscous as compared to the mycelial fermentations, like in case of fungal fermentations and actinomycetes fermentations, where the broth viscosity is going to be increased due to heavy growth of mycelia or hyphae. So in those conditions, the proper mixing or aeration conditions are going to be more focused, because if the broth viscosity increases, the solubility or volumetric airflow rate is going to be decreased, and therefore KLA is going to be affected due to this mycelial growth. Then the rheological properties of the product. Microorganisms may produce extracellular metabolites or the products that may be responsible for increasing the viscosity of broth. So all these are the parameters which affects the broth rheology. Therefore, it is highly essential. So as we have learned that broth rheology, it affects oxygen transfer and mixing characteristics. So that is, it affects the relationship between KLA. So finally it affects means what? It affects oxygen transfer, it affects mixing characteristics means what? It is going to affect KLA, that is the aeration capacity of fermenter, and it is going to affect the speed of agitation. So as we can say that as the viscosity of broth increases, KLA decreases, the aeration capacity of fermenter decreases, oxygen transfer rate will be affected. So what is important here? The important thing here is that the relationship between KLA and power consumption has to be quantitated at particular viscosity of fermentation broth. This is very important. So relationship between KLA and power consumption is quantitated at particular viscosity units, and then the required agitator speed is calculated for the large scale. This is how, so KLA has to be determined considering the viscosity of broth, considering the factors which are responsible for modification of rheological properties of broth, the factors as we have seen in the last slide. So this is how the scale up of broth rheological characteristics. But scale up in scale up process, broth rheology plays also very important, and it plays a very important role during the fermentation process. Next parameter we have to study is scale up of sterilization. So we have learned in sterilization chapter that there is a factor called as Dale factor. So this Dale factor is very important, or it is a criteria for scale up. Now we'll understand what is the meaning of this, that Dale factor is used as a criteria for scale up. For this, you need to recall the formula of Dale factor, definition of Dale factor, and what are the meanings of NT, N0, what is the value of N0, etc., etc. So one example of calculating the holding time while designing the process of batch sterilization we have already learned. So you just go through that part, then we can come back to see how exactly that Dale factor is useful for the scale up of sterilization. Scale up of sterilization means designing a protocol of sterilization at small scale, designing a protocol of sterilization at higher scale, designing a sterilization protocol at large scale, because sterilization is a scale dependent factor. So the time temperature relationship, the holding time are going to be different. For a small volume, holding time is going to be different for a larger volume. And in case of continuous sterilization, if we see that temperature factor is again variable. So it is not that only holding time is variable, but if it is continuous sterilization, then the temperature is also variable. So I want that you all should go through that part of calculation of design of batch sterilization, in which we have calculated holding time. Then we'll come back to discuss more about the scale up of sterilization and how this Dale factor is useful for the scale up of sterilization, which is going to be a very important, a very interesting part.